Okay, sorry, having to go back to my old technology. So I think hopefully you can see this. I was saying at the beginning we had 8 in total, and at the end each newly forming cell has 4. So we have halved the amount of DNA. Okay, so that was what happens in telophase 1. And cytokinesis is when the cell divides into 2. So those 2 daughter cells will have half the amount of DNA, okay, of the parents. Then we go on to meiosis 2. So the next stage, which is very similar to mitosis, okay, which makes it easier to remember, you just have half the amount of genetic material. So prophase 2. This is where the nucleus and the nucleolus disappear once again. The chromosomes condense again and the spindle fibres will form. But this time you can see you have 2 cells. These are our 2 daughter cells produced from meiosis 1. So each one has 4 chromosomes now. Okay. Your spindle fibres will reform during prophase. Metaphase 2, again, single chromosomes this time will line up at the equator. Okay, so your single chromosomes lining up at the equator just like before. Anaphase 2, the spindle fibres now contract, so they pull those individual parts of the chromosomes to either end of the cell, the poles, and these chromatids will separate from each other. So we've halved the amount of DNA at each end of the cell once again. Okay. And then we have telophase 2, which is the final stage of meiosis. Okay, here your nuclei and nucleolus will reform, so the genetic material will be kept inside the nucleus. The spindle fibres will disappear, and again you'll get a second stage of cytokinesis. So the cell will divide yet again. So it will divide from these 2. Each one will divide again across the middle there, because you've got the DNA at the poles, into another 2. So it's splitting there, cytokinesis, and you have 4. Okay, so 4 daughter cells. So that's how we get to 4 daughter cells from one parent cell. Okay, and these are haploid daughter cells. So this time we only have what appears to be 2 chromosomes here within each cell. Sorry, okay, so we started with 8 because the DNA in this case was replicated. We halved it to 4, which is what a normal diploid cell would have in this case, and now it's been halved in 2 again. So it's been 4 has been halved into 2. You've got 2, 2, 2 and 2, and 4 daughter cells in total. So I hope that makes sense. Because my technology has failed me again, as I said, but I really wanted to help you understand that meiosis 2 is very much like mitosis, but it has half the amount of DNA, and I wanted you to understand how, in this case, it halves from the original cell in anaphase 1 at the beginning, replicating the DNA, so it's got a double amount, and then it will halve during meiosis 1, and then it will halve again during meiosis 2.